Hey there, everybody. Just logging on here. See if people are around. Cool. Um, Looks like Jason's already on. I'm going to get you here in a second, Jason, but I want to set it up. Welcome, day two of our self proclaimed Science Week, which is awesome. Talking about all incredible and important things science at Trout Unlimited. And today we've got a really cool week. If you haven't looked at the blog yet, it's got the whole schedule of what's going on for the week. Yesterday we had the science director, Helen Neville, on to talk about kind of the general overview of why this is important, why science is important at Trout Unlimited, and the ways we use that. I was super stoked to hear her talk about the things she loves, and she's just got an insane knowledge of not only Lahontan cutthroat trout, but all things science across the board at Trout Unlimited throughout the entire country. Today, we are talking with equally as cool, who I'm stoked to reconnect with because it's been a little over a year since I've seen him, Jason Barnes. I spent time with Jason. I had the privilege to spend time with Jason last year on several different occasions. I shared a dirt floor and a wall tent out in the remote deserts of Oregon, southeastern Oregon, with him last year, trying to tell the story of the Lahontan cutthroat trout and the genetic diversity program they're working on. I'm excited after this talk that we have. I'm going to let Jason on here in a second to give us his own words, but I'm excited to talk a little bit about the release of the film that we have coming out that tells Jason's story and others within our science program, from Helen and him and others, about what they're doing in the deserts for Lahontan cutthroat trout. So without further ado, let's get the man on. Here he is. Let's see if he can. Here, I just sent you a request, Jason. So we're waiting for him. Last I saw Jason, there he is. Yeah. What's going on? What's up, boss?
Not too much. Just pretending to be civilized for a short time here, maybe an hour.
You're looking well groomed these days. So,
Yeah.
not living out of a wall tent.
Not living out of a wall tent. No, I did that for about a month in September, and I'm back in civilization. I was just in Montana and Idaho and Wyoming, as you know, and I was a little uncivilized there, but more civilized than normal. So,
Yeah. It helps to kind of press the reset button over there, I bet, huh?
it does, especially after a summer staying home mostly and watching my field crew do all the good work.
Yeah, I know that. I can tell that probably pains you a little bit at times. I just want to give a special shout out to Jason Barnes, but also Gene Barney for this sweet LCT mug that I'm rocking right here. It looks like you got one behind you as well.
I got mine too. I was going to give a shout out to Gene for that too.
Hey man, we're thinking the same thing. I use this often, so Gene, if you're out there, thanks for the midday coffee break here.
Absolutely, and it holds so much coffee. It's perfect. It's a long day at home.
It is huge, man. Um, yeah, so I was just kind of setting this up. When I met you this time and a little bit before last year, you picked me up pre-COVID days in the airport, and you shuttled me out to your site that you were working on, and it was in the middle of freaking nowhere. Pretty cool country over there too, in southeastern Oregon. Not exactly. I mean, I guess it's what I expected, but gosh, man, you were just saying, you spend a lot of your career living in the places that you're studying, that you're working in. You not only just talk the talk, you walk the walk with your field crew. You're right there side by side, and I got a deep appreciation of like how y'all interact. And so maybe let's start there. Talk a little bit about, like, you said this field season's a little different, but I really want to set this whole thing up. You guys head out into these places and you live it and breathe it every day, and you're working not 8-hour days. We're not punching a clock at the end of the day. it's the whole time. I mean, 24 hours a day, rather, you know, and from the minute you step foot on the ground there, you're thinking about what your objective might be. And I guess for this particular project, um, talk a little bit about that, like what you guys had set out to do. And I just gave people kind of an update. There's a blog out there, great blog that Jason wrote that we posted today if you want to get some more background, but I want Jason to bring it to life a little bit. So talk a little bit about how your project came to be and what that looked like.
Okay. Well, I mean, I think, aside from what you're going to read in the blog, maybe the stuff that you don't get to see is what really went into that whole project. And, Helen and I started planning that project in January, and we had planned that project for months. We were ready to go by the time we got on the ground. All the logistic considerations were ready. And we were standing on top of this mountain, looking out over this range, and we were looking at a map and realized that most of it was on private property and that we had not contacted that landowner yet. And it was, me and another guy on my field crew, Jason Schumacher, we stood up there and we considered whether we went ahead with the project or whether we did it the right way, and went and contacted the land. And because we had permission to access the land from the state agency, but not from the landowner. So when we were up, we decided, hey, let's do this the right way. Let's go out and let's go talk to the rancher. And so here we are, truck loaded, ready to go for 2 weeks of work. And we go and talk to the rancher, and he says, absolutely not. You guys are not allowed on our land. You're not allowed to do the study. And so, the 5 or 6 months that led up to that turned out to be all for naught. But in typical fashion, in my work for LCT, like, I just don't give up. And
No for an answer.
Yeah, so I had a super busy, it was a month and a half or 2 months. I was on the road sometimes 20 hours a day, sleeping on the side of the freeway on the way to my next spot going. I think I drove something like 2,500 miles that week. But I stopped. I don't even remember where I stopped. I think it was in Elko. I stopped and I picked up a bottle of whiskey. And I said, I'm driving to Oregon and I'm going to go try one more time to get him to say yes. And I drove out there by myself, just dog tired, and had a conversation with the rancher and just convinced him that this was in his best interest, which I still believe that it was. The partnership with you and a rancher, I think, is one that can pay dividends in the long term. And, 2 months later, finally got a yes. I drove all the way home, which is, 6 hours after being on the road for 20 hours. I told my guys, like, let's get packed up. We're starting tomorrow. And sure enough, they were ready to go. And we were out there trying to make up 2 lost months. And, only with the crew that I have with those guys and their, amazing work ethic and their hard work. And they genuinely care about Lahontan Cutthroat Trout too. So, they were out there putting in, 12, 14, 16-hour days doing the work, trying to make up months of lost time. And as you know, the schedule in September to move those fish got pushed back 2 weeks. But after 2 months delay, we're 2 weeks behind. I feel that was a testament to them. But yeah, there we are, and it got done.
Yeah, and so I guess for some background on some of the stuff that you do, your primary focus is on Lahontan Cutthroat Trout, and I saw. I've seen a bunch. You're obsessed with them. Let's put it that way. Jason also, in case y'all don't know, if you look behind him, couple pieces of his artwork there. He's an accomplished photographer, painter, artist as well. So this guy lives, eats, breathes, sleeps, draws, paints, documents everything about these fish. And I learned a lot by watching you and how you study the habitat and what you're doing. And that actually oddly benefits me in the photo world too. But talk a little bit about this project. We're talking about genetic diversity of trout. And when people approached me, or when we came up with this plan to document this is a really wonky topic, but like it's one that's very important, not only to you, the work you do, but it has implications across the country and maybe in some other work that we do as well. So talk a little bit about the focus of genetic diversity on Lahontan Cutthroat Trout and why that might be an important thing to work on.
Yeah, I think for any fish, when you're considering them from the biological perspective, that genetic diversity is important. That's basically the tools in the toolbox that those fish have anytime they need to adapt to a certain scenario. Um, but for Lahontan cutthroat trout, it's especially important because historically they lived in these large, what are called meta populations, or a collection of small populations that intermixed occasionally. And in the last 200 years or so, there's been a lot of development, whether it was mining or ranching or even paper mills that disconnected these habitats. And so these fish quickly, living only 6 or 7 years in a stream, they quickly start to lose diversity, and then their toolbox becomes more and more empty. And what we were left with was a trout on an incredibly arid landscape that's always changing, with basically a screwdriver in the toolbox. And so this study that we're doing, the genetic rescue study, is basically trying to reconnect populations that were once connected. And in those isolated populations, each one of them may have a different tool in that toolbox. And so basically, we're just trying to get them all back together so that they can share those tools and be capable of surviving whatever the future may hold whatever the future may hold, climate-wise or reality on the ground, whether it's diversions. And as I said in the blog, what we'd love to do is go out and reconnect all these populations. It would be awesome if all of these river populations were connected to their tributaries again, but that can take on the order of decades sometimes. And with so few populations out there of LCT, we really don't have the time, because these fish are continually losing those tools out of the toolbox, or losing that genetic information. And so we needed to figure out a way that we could go out there and, with the help of humans, selectively move these fish using probable connected populations, or ones that we knew. And where we ended up doing the study was the perfect place, because they had stocked 7 streams with Lahontan cutthroat trout from 1 or 2 streams, and they had been isolated for 40 or 50 years. And so basically all we're doing is reintroducing that connection, and then we're trying to see what happens. And that would mimic, what may be a tool in the future for managers to go out and conserve any populations they have. Because even though it's especially important for Lahontan cutthroat trout, genetic drift affects every kind of living organism that there is. I mean, once you're isolated, it's completely and totally possible to lose genes that aren't selected for, or even ones that are selected for that are rare. So this really is an important study, and it could pave the way forward for a lot of different fish. And they are using it with brook trout back east in one small population, and shown some pretty promising results. And we're hoping to see the same thing with Lahontan cutthroat trout.
Right. And like, this is not your bucket biologist type thing, man. You guys, I watched you section off multiple streams with nets, catch nets, and you guys not only study the fish, but you study the habitat around it. You guys are diligent in every facet of what is going on with these fish out there. And one thing I took away from your blog when I was reading it is that I found interesting that you could maybe expand upon, was like, one of your quotes was like, we look at where a species is now rather than look at how it got that way. And I guess I'm interested to hear your take on that and what that means exactly.
Yeah, so if you guys saw Helen yesterday, you know that she's our senior scientist, but she's also my mentor, and we have a lot of discussions. And I do a lot of reading on like the historical and the last 40,000 years, the last as much record as I can go back. I read as much as I can on there, and it's really important. I think genetics. If you take a genetic sample today, what you see is a snapshot of where the cutthroat are today. And what I try to bring to the conversation is where they've been, how they got there, and how that might inform the future. And that was a big piece of what I did. Was to kind of reconstruct the evolutionary history of this fish. And I didn't mention it in the blog, but I beat the academics to that whole finding by at least 5 years, just because I was reading outside of the fish literature. Because
Right.
I'm insanely interested about the Great Basin. Like you said, I live out there, I live and breathe it. I'm interested in more than just the fish, because the fish is kind of an iconic piece of the landscape, but it's the landscape. Being a native Nevadan, it's what I've grown up with, it's what I've known, and I'm really passionate about it. So going out and reading that obscure literature, finding those old reports from the 50s and 60s, digging through the state wildlife agencies file cabinets and grabbing out those dusty old folders, like that stuff really gets me excited, because there's a lot of information. There's not a lot of information on Lahontan cutthroat trout, but there's more information than some people think might be available. And so that's kind of been my mission, is to dig a lot of that up and to see what we can learn, because for the last 50 years since this fish has been listed, we've been making a lot of the same mistakes. Right. And I think that our role as Trout Unlimited is to focus on the science, but then also to dig deep and to help the agencies figure out how we can maybe learn from the past and move forward without having to trip all over ourselves and make the same mistakes again. So that's a big part of reading into the past, and then trying to get that to share that with how we move forward.
Right. And I think, that's what makes TU so uniquely different in a lot of ways, is we have that capability to kind of think outside of that box and kind of, trust the work that you do, and know that, we can go beyond the inner circle and see, the whole picture. And I can't express enough how uniquely cool that is, about these fish and what they're doing. And I don't think I know anybody that's doing more for these fish than yourself and Helen and others, partners that you're working with out there right now. And I guess, a question I had, we talk about, these fish, you've studied them, you've located, and if you read the blog, like, it's a really cool picture of what's going on. And then eventually watch the film, like, you study these fish and you realize that there are certain populations that you're trying to find high and low genetic diversities and things like that. And without getting too much into the weeds, I'm always curious, like, how do you guys do this? Like, how do you understand, like, what fish had the right set of genes to infuse with the other set you're not making a fish, essentially, you're just diversifying the fish, correct?
Right. Yeah.
So how do you guys determine that?
Yeah, so luckily for you, I'm not a geneticist, so I can't go deep into it. But Ellen did, and there is some historical information that showed that the population that we moved the LCT from to the population we moved LCT to had a different suite of genetics. There were a few genes or alleles there that were different, that we thought we could kind of use as markers, and we didn't know if they were necessarily beneficial. We just knew they were different and they had been lost to those populations. And so that's basically how we selected it. Is we knew that was the source population 40 years ago, and we knew that they were different. And so that made just the perfect study system. And, the way these streams are set up, there's 7 streams that initially we were going to study. We ended up with 4. But 4 of those studies all drain off the same side of the mountain. So they have the same aspect, they're getting the same sun, they have similar temperatures, similar habitat. It just couldn't have been a better spot. So that's kind of why we chose where we did. We went through a lot of different scenarios before we got there, and then this one opened up, and it just happened to be perfect.
And then in September, like you had mentioned earlier, it was go time. I mean, like, almost rather quickly, you guys had to turn around. You had these fish that were selected for the project in a holding space in a stream by where we were camped, and then it was time to move them with the help of the agency as well, and bring them to these streams that you guys have documented and put them in there. And I think, how do you know? I guess how will you know? I know this year was different because of COVID, but like, how will you know if the infusion worked, if these genes took hold, if it makes a difference, basically?
Yeah, so I mean, when you think about this from really simplistic terms, there's a lot of people that go out and they put cut bows into systems, and they put cut bows in there because those hybrids get big. And right. This is kind of no different. So even though they're not hybrids, it's an LCT crossed with another LCT. There is new genetic information in those fish, and so what we expect to see is that those fish that are coming from that LCT cross between the distant population and the local population, those fish will actually be bigger, and that's exactly what we hope to see from across the range. Because there's a lot of people that complain about LCT's small size, and one of the things that I always tell people is that well, one of the reasons they've lost so much size is because they don't have what they need. They don't have connection. But we have seen in places like Maggie Creek where they've reconnected these populations, like Coyote, Little Jack, and Beaver Creek. The fish got bigger, and they were catching 24-inch cutthroat down in the main stem areas. And so that's exactly that's one of the things that we expect to see is when we go back and we look at these fish, we should have these graphs that show some larger fish and some smaller fish. And then when we go to test the genetics, we'll see if the genes of those larger fish tend to be the quote unquote hybrids. I mean, it's cutthroat by cutthroat cross, but so that's one of the metrics. And then the genetics also will show, because they've got that distinct allele in there, we'll be able to track which population came from where.
So you'll go and resample the streams and measure it that way, I presume, right?
Yeah, I mean, that's the idea, just so there's no bias there. We won't call anything just based on its size, but
right.
That's things that we'll look at is if we see the signature of those other LCT in the source and the donor populations, and then we see bigger size, we'll see that not only did the experiment work, but the outcomes came out exactly as we hypothesized.
Yeah, man. And like, I was thinking about it too, on the small fish scale, like, these streams, the way you worked it was, through an electrofishing technique. And there were some fish in there that I saw that were not small fish by any stretch, and a bit of a surprise. I mean, like, they're the boss of the hole, and there's some decent fish. I mean, it kind of blew my mind to see some of the bigger, I wouldn't say 24-inch, definitely not, but, bigger than you would think for the stream size that we're in. And these are not easy streams. I mean, granted, they're all on private property, so they're not fishable, but if you were to even try to fish, these are not easy streams to get into. They're rugged, they're remote, they're steep, they're drops and ups and down and pools and rock and sagebrush as tall as me, and it was burly. I mean, it was crazy.
Yeah. And I think that's a testament to these fish too. That really kind of roughly explains the character of these fish. They're able to get huge in these small systems, and we try to tell that story to people, but because there's basically one population that has everything it needs, that's all on private property, we don't get a chance to showcase that to anglers. And
right.
There was comments earlier about, like, well, how do you fish for these things and where do you fish for them? And right. I think an important to people is that there's not that many populations left. Most people know Lahontan cutthroat trout from Pyramid Lake, but they exist in a ton of other places. It's just that most of those aren't easy to fish, or they're all on private land. And most of the water in Nevada is on private land because it's a dry area, and those ranchers and farmers came in early and bought up land in the valley bottoms. And fortunately, we've had some great ranchers who have cooperated and not only worked with us and others to have Lahontan cutthroat trout, but we're genuinely excited about getting those fish back on the landscape or maintaining them as they had in some cases.
Yeah. I got a question from a viewer here, and it just mentions, like, and I think we touched on this a little bit. How are you conducting these DNA detection samplings? I mean, you guys are taking these and sending them off to a lab, right?
Yes. Yeah. We're not doing any of that sampling in-house. We just take a fin clip, and then they're going to, I believe it's the University of Nevada. That's where we drop the samples off, is here in Reno. And then they're just doing shotgun sequencing on those genes to look at more of the genome than just using other methods.
And then somebody asked this question. I don't know quite what it means. Do you have any system for reporting LCT populations for rarely fished waters? And I'm guessing that means more of like a community scientist or an angler scientist kind of approach? Like if somebody discovers it, maybe, do we have a way that people can report these, or that, is listed somewhere?
So most of the populations are listed, in the respective state agencies' fishing books, whether it's California, Oregon, or Nevada. Um, but there are some people who go out. I know of one or two people who are constantly out there fishing these no-name streams in the hardest reaches looking for these fish. And those
Right.
people are in contact with TU, they're in contact with the agencies. So no formal reporting of these rare populations, but I can say that from my perspective, every one I've ever heard of, I followed up on. In fact, the last one that I heard um, was one of our own within TU told me about a lost population of Lahontan cutthroat trout in Yosemite National Park.
Wow.
And so I went through and did a whole bunch of the research. And this year I was going to take a vacation in the middle of the summer and go up there and try to find them, because one of the things that we're struggling with is such a loss of genetic diversity. And so I thought, well, man, if I could go up there and get some fin clips off these fish, 10 years after they were last seen, maybe those represent some genetics that have been lost to the species as a whole. And if
Right.
this rescue project turns out to do what we think it's going to do, maybe that's a way to reintroduce genes into these LCD populations that are struggling so much.
Yeah, and this viewer was saying the same question that he asked. He said, he's caught some of these creek run LCTs that might have that info, and he'd happily get it to you. I'd say, Jason's pretty open cat. I would send him an email if I were you, and maybe just hit him up. Yeah, I'm sure that's okay, right?
Oh,
If he,
absolutely. Yeah,
yeah.
I feel sorry for people that email me sometimes, because sometimes they force me to geek out and read a whole bunch, and then I just puke out a whole bunch of information on them. So just be prepared.
Right. I guess I wanted to pivot a little bit because you mentioned it earlier. You and I have had several conversations. I've been out to, and a lot of people I think watching is have been to Pyramid Lake. They've seen those giant fish. They've seen, the way people fish for them. It's cool. It's definitely big cutthroat in a desert lake. Talk a little bit about that and versus these fish. I think a lot of people know LCT from this lakes, but what they don't know is that there are these populations that we're studying too. And I guess maybe speak to the Pyramid Lake versus other populations as well.
Yeah, so like you said, most people know LCT from Pyramid Lake, and I think that we should be eternally grateful for the fact that Pyramid Lake has brought a lot of attention to LCT. And that attention, whether it's translated into conservation work for the other species or not yet, is kind of irrelevant, but it's definitely brought attention to them, and now they're on people's radar. But I think, this study and then other work that we put out has been a really important source of information to tell people that Pyramid Lake's not the only place where these fish exist. And even if we did recover LCT in Pyramid Lake, it wouldn't take them off the endangered species list, because it's just one piece. It's one of 3 lakes that has LCT in it. The other 2 are one of 4, I guess. But there's only 2 lakes where these fish naturally reproduce. And, we've been trying to stay under the radar and not draw attention to them. But it's also the case that we need people to care. And there are a few dedicated people out there who care about these other lakes that have held LCT for all these years. And they're dedicated to protecting them. But, Pyramid is one lake. There's 73 stream populations and, a handful of small lakes. But Pyramid Lake is, it's a hatchery supported lake. And it does provide an opportunity to go after these giant fish. But most people that fish there, they hear it as a conservation story. I remember I was quoted in that Al Jazeera America article that basically was like back from extinction. And thank God I was misquoted so that people don't associate or not misquoted, but I think they called me Josh Barnes in the article. I'm okay. I hate to be associated with it. Because extinction is forever, like that's
Right.
what we're facing. species. And to say back from extinction is like, it's not only a bastardization of the term, but it implies that LCT were extinct and now Pyramid Lake has been the savior of the entire species.
Right.
And you think about the fact that they just spent $34 million on this passage. Like, that's wonderful. It's great that they were able to do that. But we're scraping together every dime that we can get to save the rest of the LCT in Eastern California, Southeastern Oregon, and most of Northern Nevada. And I mean, we don't have near that kind of money. And, you know, there's some long-term implications of the recovery of Pyramid Lake, because the Truckee River feeds into Pyramid. And the Truckee in and of itself is probably the greatest fishery in the whole state of Nevada. I mean,
Right.
I've caught 30-inch browns in there, and you'll never find a bigger proponent or supporter of LCT. But unfortunately, the reality of recovering Pyramid Lake haven't fully been dealt with, because there's just been such a lack of information that's been put out there that I think most anglers don't even know, you know, what could potentially be coming for the future of the Truckee River.
Right. Right. That's very interesting. So, you know, what's next? I mean, are you guys, what's the next steps here with this project? You know, and the film that we're going to release here in about a half hour talks about what's been, and then I guess my curiosity lies in what's to come, and what the implications for all this mean down the road.
Yeah, so the next step is to basically redo everything we did last year. You know, we've got a method of population sampling that, you know, thankfully Dan DeWalter on our science team has provided us with a really good template on how to sample these populations and get, you know, fairly accurate numbers.
Right.
But the unfortunate thing is it takes a tremendous amount of work. And so that's why we're out there working 12, 14, 16 hours a day, is because we need good numbers, because we want to be able to detect a small change in these populations. So we need, you know, narrow confidence intervals. We need to, you know, characterize these populations exactly. And like you were saying, going out and sectioning off these streams, it takes a lot of work. You know, you put up block nets, you electrofish, and you go through as many passes as it takes to get all the fish that you can get down to a selected criteria. But that basically took, you know, 2.5 or 3 months of solid work. And, you know, we came up to the deadline and my guys were like, we're not going to finish in time, so we're going to work extra days. And I'm thankful that I have like such a dedicated crew.
They're rock and roll.
They're pretty amazing.
Yeah.
And so, you know, we're going to go out and we're going to sample the population again. We're going to see if it got bigger. Because one of the things about the place where we did this study that we're not fortunate enough to have elsewhere is we're not habitat. Habitat in the place that we did the study is incredible. And that's not what's limiting these fish. If anything's limiting these fish, it's probably going to be genetics, which is another reason that we did the study there. So we're going to go out again and see if those populations have expanded. We're going to clip the caudal fin of all the fish that we get our hands on, and we're going to run those genetics. It's funny because, you know, all the work we do, all the hours we spend, most of the money is spent getting the genetics run.
Right.
So we're cheap labor compared to those guys in lab coats. We're getting dirty and fun, so I guess that's our payment. But yeah, we'll clip as many fish as we can. We'll send those in for genetics, and then we'll test them again and see if, in fact, it did have a positive impact on those populations. And we do have one control population where we've sampled it thoroughly, and we've got 3 test populations that we sampled just as thoroughly. But that control population, we didn't move any fish into. So we'll be able to see, you know, if it was a good water year or an average water year, which can sometimes be better, we'll be able to see the effect of the genetics outside of just putting those fish in. Because we didn't remove or put any other fish into the control. And so we'll control environmental impacts and be able to see the difference between, you know, what we did in those other 3 streams and where we didn't do that.
Somebody just asked if you guys take any volunteer workers to help with that. Is that a thing, or No, or you guys plan on it in the future? Oh, paused out there. Can you hear me? Still there, Jason?
Still got me there?
Yeah, I'd say just reach out to Jason. And yeah, I think you cut in and out. I don't know, but you're back now.
Okay. Yeah, the short answer is yes, we can use volunteers, but like you said, we're so remote and our trips are so long that as long as you're willing to go out there and kind of support yourself, we're happy to have people and show them these fish.
Right, right. Cool, man. Well, I'm stoked. I'm stoked to share this story. I hope we can continue to tell this story down the line when you find out some results and things like that. So plan on keeping us updated, Jason. Maybe we'll talk, in a year from now and figure out, what's been added to this, how the project has worked, and I'm excited to hear about it personally, and I'm sure others on this channel are equally as curious about it. And I just want to say thanks for your time and effort. Thanks for letting me put a camera in your face for a couple weeks and all that good stuff. So it was quite a trip, man. I mean, again, sleeping on the dirt and hanging out and hiking our butts off and working long hours. It was all worth it. So it was very cool.
Yeah, and thank you. I mean, I've been fortunate enough to see the video, the film, and I hope that others get a chance to see it too and kind of get a sense of what we're doing, where we've been. You did a great job, so thanks for getting that story out there.
Oh, anytime, man. Well, I just want to give a quick update, folks. We're gonna push live on this thing here in about 20 minutes. So give it a look. It's either it's on our YouTube page, but it's also in the blog. We have links to it in Jason's blog from today. You can sign up for a quick reminder that when it goes live here shortly, and looking forward to it, man. So again, thank you, Jason, for your time and effort. You guys are awesome. Thank you to the crew you work with. Those folks are amazing, passionate, and interesting individuals that just love these fish. So excited to hear more. So thank you again, Jason.
Yeah, thank you, Josh. Can't wait to give the update.
All right, man. Take care.
See ya.
All right, later, boss.